Welcome to today's VitisGen 2 webinar, in which we'll be learning about flavor challenges of using North American Vitis species when breeding grape varieties. My name is Janet Van Zorn, and I'm part of the Extension and Outreach team for VitisGen 2, as well as an Extension Support Specialist for the New York Statewide Viticulture Extension Program. A few comments before we begin the presentation today. Our next webinar in the series will be in the fall. To stay informed of upcoming webinars, as well as to learn about new publications, accomplishments, and other information about VitisGen, you can follow us on Twitter at VitisGen2. You can also visit our website or subscribe to our YouTube channel to see previous webinars, and those URLs are shown on the screen and will be also provided in the chat a little bit later. So speaking of, if you move your mouse to the bottom of the screen, you'll see a few icons. You can click on chat to open the chat bar and use that to introduce yourself, make comments, or to alert me if you're having technical difficulties. Please just change the chat settings to send to all panelists and attendees so everyone can see your introduction. Another icon on the bottom menu is the Q&A bar. And you can use this space to ask questions. So we'll have time for questions at the end of the presentation, but feel free to type them in at any time during the talk. Our speaker today is Dr. Gavin Sacks. Gavin is a professor in the Department of Food Sciences at Cornell University, and his lab provides chemical analyses of grape flavor compounds for the VitisGen 2 breeding populations. In particular, his research focuses on compounds responsible for the flavors found in wild Vitis species. And with that, I'm going to turn the screen over to Gavin.
Great. Thank you very much, Janet, for the introduction, and thank you to Tim for, let's see here, at this point, I'm going to share my screen. You should be able to see it. Janet, did it pop up?
Not yet.
Okay, let me try it again. Oh, oops, missed the button. Now?
Yep.
Okay,
there we go.
Great. And thank you both to Janet and Tim for organizing these webinars. I'm glad everyone can participate regardless of location or how you're dressed or without you need to be checking email in the background while you're doing this. So today's talk will be about the flavor challenges of using wild grape species either to make wines from wild grapes or more commonly to make wines from the interspecific hybrids of these grapes. So what I'm going to talk about first is the variation that exists amongst grape species, and when we say wild Vitis, and give a brief overview of what VitisGen 2 is and what we are doing with the project and my specific role within the project. And then I'm going to take a look at the flavor challenges with wild Vitis from a few different perspectives. One is the perspective that's easiest for me, which is the chemistry side. What's the cause? How do we relate that to sensory changes? Then I'm going to talk a little bit about what we can do now, either in the vineyard or in the winery. This will not be an exhaustive treatment of all of the approaches for dealing with these challenges, but give some idea of what you can do if you're currently trying to produce wine from interspecific hybrids. And then finally, how we're moving forward, how we're working with grape breeders and molecular biologists to understand at the biochemical level why these differences exist, and to see if we can facilitate the breeding of new varieties that lack these challenges. Okay, so very quickly, Vitis is a genus. It describes a number of grape species. The one you're most likely going to be familiar with from a wine appreciation course is Vitis vinifera, also called European wine grapes. This is the major species worldwide, and I have to say worldwide because at least here in the Finger Lakes or in upstate New York, that's not the case, but worldwide it is the major grape used for wine production, also for table grapes and in many places for juice grapes as well. But there's dozens of Vitis species known. Some of these are used or have been used historically for producing new grape varieties. As Tim Martinson is fond of pointing out, it's not appropriate to call these hybrids since all grapes are hybrids. Cabernet Sauvignon, for example, is a hybrid of Sauvignon Blanc and Cabernet Franc. Both of those are vinifera parents. So to be accurate, we should call these interspecific or interspecies hybrids. You will find me getting sloppy and using the term hybrid to describe interspecific hybrids, and I'll try my best not to. But generally, in this talk, I'm saying hybrids, I mean interspecific hybrids. That is, grapes that have both wild vitis and domesticated European wine grapes in their background. And so within these wild vitis species, outside of vinifera, the two general classifications, and this is a chemistry approach for classifying them as opposed to a genetic approach. But on the chemistry side, they tend to fall into one of two categories. There's large berried wild vitis species, which would include Labrusca and Labrusca type grapes. And the best known of these would be a hybrid of interspecific hybrid of Labrusca and vinifera, which would be Concord, and Niagara would go into this category too, along with several other grapes, Messingensis, rotundifolia, which is not strictly speaking in the same, or muscadine, not strictly speaking in the same genus. These are characterized, if you've ever had Concord grapes, they have large berries. They have very thick skins that slip off. They're often called slip skin grapes. They tend to grow down or sprawl. They don't always have a lot of color. Sometimes they'll have some purple, but they can exist in bronze or other less dramatic colors too. And they're characterized by so-called foxy aromas, which would be some of the characteristic aromas of purple grape juice and white grape juice here in the United States. It's the aroma of, say, a Jolly Rancher grape flavor, or in the case of some other grapes, the aromas of caramel and cotton candy. There's also small berried wild vitis, and at least here in the Northeast, these are the types of wild grapes that we most frequently encounter, say crawling up the sides of telephone poles or alongside riverbanks. And a few examples, Vitis riparia, Vitis rupestris, Vitis cinerea, I think it's riparia depicted there. And these are commonly used in breeding programs, and one of Bruce Reich, our grape breeder here, Cornell's most recent releases, Arundel, has all of these, I believe, in the background, but particularly riparia in the background. These are characterized by having smaller berries, so more akin to Vitis vinifera, upward growth habits, thinner skins, much more striking, darkly colored fruits, and generally have little aroma or some herbaceous aromas, but they don't have the strong, foxy, grapey aromas of the larger berried varieties. And most of the time when these are classified, things with large berried backgrounds are often referred to not as interspecific hybrids, but rather as so-called native varieties. These small berried Vitis interspecific hybrids are referred to as hybrids, or sometimes French-American hybrids, if they're some of the classic ones, or just interspecific hybrids. These are the commercially grown cultivars are almost always hybrids between Vitis vinifera and other non-vinifera species. So the reason these are of interest to grape growers is that Vitis vinifera is notoriously disease susceptible. It's also more subject to environmental stresses like cold damage. I saw there was a conversation going on the chat room before the talk started about winter lows in the upper Midwest. Vitis vinifera would have a hard time surviving. Even if it survived, you wouldn't have very much fruit in those kinds of challenging conditions. Furthermore, if you try to grow Vitis vinifera without any kind of spray program in humid environments like what we have, this is someone's attempt at no spray vinifera. This was part of an experiment we were doing on the effects of sulfur sprays, and we needed a control. And by comparison, you can make interspecific hybrids using wild vitis with natural disease resistance that has excellent cold hardness, that will need to know, or at the very least, fewer sprays in order to maintain healthy fruit. So the problem with using, or the challenge associated with using wild vitis, is associated with its organoleptic properties. So if you've had the experience of trying to eat some of the wild grapes that you can find growing up the size of telephone poles, one of the first things you likely noticed was the sourness of that fruit. And if you turn these wild vitis into wine, which we have done, the wine will often have a thin mouthfeel. That is, if you can get past the sourness due to a lack of tannins or astringency in the wine. The wines produced from wild species or their interspecific hybrids. The large berry species are characterized by their foxy aromas. This is perfectly acceptable if you're trying to make Concord grape juice, or if you're trying to make a wine from Concord and you have a consumer who likes that kind of wine. It's less acceptable for most red table wines or most white table wines. Small berried species are characterized often by having herbaceous or vegetal aromas, green aromas. And finally, one topic I won't be getting much into detail about today is color problems. Wild grapes are characterized by having higher concentrations of anthocyanin and diglucosides, that is, anthocyanin pigments that have two sugars on them. These forms of pigments are not able to make more stable forms during winemaking, and so they tend to suffer from color loss more rapidly. So, the idea behind VitisGen, which is a large multi-PI, multi-institution grant funded by the USDA as part of their Specialty Crops Research Initiative, is to try to facilitate the more rapid development or more efficient development of new grapes that are going to bring along the desired properties of wild vitis, so cold hardiness, disease resistance, but without dragging along the undesirable properties. This is no different than what breeders have been doing when they've been making crosses for, well, formally for the last decades or centuries, but going back even further than that, just by chance. You want to get good growing properties, you want to get good flavor. What we're trying to do is to use modern analytical tools and genomics to try to make this process go faster. And through this, then, to try to realize some of the advantages that have been seen in other crops. So this is data. It's unfortunately not resolved all the way what the units are, but it's bushels per acre for corn, and the effect of genetics and technology in the yield of corn over the last several decades. This same sort of increase in productivity or in diversification of flavor or in decreasing the need for sprays does not exist across for grapes. We're for the most part still growing, planting, making wine from the same varieties that were grown for the last several centuries. And one of the goals of this project is to try to overcome some of the concerns associated with bringing in new varieties, which is that you want them to deliver high fruit quality in addition to desirable growing behavior. Okay, so the way this would look in practice, we have several grape breeders who are members of this project, including ones at Cornell and at the USDA, both in California and in New York State, in Minnesota, in South Dakota, in Missouri, and I suspect that I might be leaving some people out, unfortunately, but I think those are the main collaborators on the project. They will have populations of grapes, for example, mapping populations which have the same parents, but are different individuals. That means that they are all siblings of each other. So across a mom plant, a dad plant, get a bunch of brothers, get a bunch of siblings, and you can do a genotyping of the population. That is a genetic characterization to see where they differ genetically. And then this is where our team gets involved. We don't do the genotyping side. We're interested in the chemotyping side and understanding chemically what makes these different sibling grapes different from each other, and in particular attempting to measure what chemical traits differ that would relate to flavor properties. For example, malic acid is one of the key contributors to sourness, so we can characterize that in this population. And we see, even though they're the same parents, just like you'd expect to see differences in brothers and sisters in a human family. You can see the same thing in a grape population as well. And then we can combine the two together and understand which genes, it's often plural, which genes or genetic regions are associated with having a particular trait like high malic acid. And then the long-term goal is for breeders then to go to new crosses they've made, new individuals, and decide which ones they want to keep and which ones they want to get rid of based on their genetic profile. So instead of having to make a cross and then wait several years before you have fruit to test or do sensory evaluation on, you can do this immediately. And you can make sure that as a breeder, the only things you're saving, putting into the field or into a greenhouse, are the most promising candidates for delivering high fruit quality cultivars later on. And so put another way, we're trying to make the pipeline more efficient. We want all the grapes that are, or as many grapes as possible that are being investigated by grape breeders to have a high potential to deliver high fruit quality. Okay, so today's talk, we're going to go through these challenges one by one, except for we won't be going through color, in part because our particular team is not working on it all that much. The genetic region associated with diglucoside production has been characterized, and so we're working with breeders to help them figure out if that existing marker or existing genetic region works to their populations. But where we're focusing a little bit more is taste, mouthfeel, and odor, because of the we're interested in both understanding chemically what's creating these differences and figure out efficient ways to make those measurements. So getting started first with taste problems, and in particular sourness. I put up lemons for a reason. The concentration of titratable acids, so if you do a titration experiment on grape juice, a typical titratable acidity in a grape or in a wine is somewhere around 0.5% by weight, maybe up to 1% occasionally, 0.5 to 1%. By comparison, a lemon is more like 5%. So if you think that a lemon is more sour than a typical grape, you are correct. A typical lemon is more sour than a typical grape. Wild grape species routinely have 2%, 3%, 4% titratable acidity, even when they are mature, at least based on a sugar from a sugar perspective. So the the main taste issue associated with wild grapes is often the fact that they have very high titratable acidity. And then along with that, they can also have high pH, which is counterintuitive. So what I have here is data cribbed directly from Tim Martinson. Thank you very much for sending this raw data along. You and Chris Gerling have been collecting information on grape samples, sugars, titratable acidity, pH, and other parameters as part of Varieties in a Harvest, the excellent newsletter you put together. And so we took this data over several years and plotted out. So each one of these points is a different grape sample, different grape variety. And there is a modest correlation, as expected, between titratable acidity and pH. So when you've got high titratable acidity, more acids, on the average you have a lower pH, which makes sense, although there is some variation, and we'll talk about why in a little bit. Typical targets for winemaking are going to be for somewhere in the range of 5-7 grams per liter of titratable acidity as tartaric acid. There's going to be examples of wines, extremes on either side of that. For example, sweet wines often will have higher titratable acidity because it's balanced by the sugar, but a typical dry table wine, these are normal targets. pH, typical targets are less than 3.6. Once you get much below 3, it's a little challenging because then that results in high molecular SO2, and it can make it such that the wine is unpleasantly it has an unpleasant aroma or character from the sulfur dioxide in wine. So a typical target would probably be somewhere between pH 3, pH 3.6. And the limit on the upper end for pH is, as the pH goes up, spoilage potential goes up in the wine, and the other thing that can happen is you can see changes in wine appearance, wine color. The intensity of red wine color decreases. The wine often looks muddier and less reddish purple. There can be some undesirable changes during wine storage at high pH as well. So typical targets somewhere in this range, pH less 3.6 or less, TA somewhere in the range of 5 to 7, and these constraints are in opposition with each other. And the reason is that, say you have a Riesling at a lower pH, titratable acidity of 8 grams per liter, this would be a pretty normal pH and TA to harvest Riesling at. Riesling is a viniferg grape. The titratable acidity might be a little bit high, and the wine will be a little sour, but you can easily adjust the TA down through addition of carbonate. Same pH, almost double titratable acidity, this Vignoles grape, which is a interspecific hybrid. Very high titratable acidity of 17 grams per liter. This wine, if it's turned into a this grape turned into a dry table wine, will result in an unacceptably sour wine in most cases. You can bring that pH you can bring that TA down through addition of bicarbonate. When this happens, the pH goes up. You're adding a base, you're decreasing the titratable acidity, pH goes up. By the time you're done, the pH will often be well over that benchmark of 3.6. It might be 4.0, it might be 4.2. You can still make wine and sell wine, it just gets harder. It's much easier to have spoilage issues and other problems once you get that high. So what you run into, so one of the main problems that winemakers run into when they use wild vitis, if they're trying to make a wine from wild vitis, or more commonly from interspecific hybrids, is that on the average, interspecific hybrids have higher pH and higher TA. And the way that you end up with both higher pH and higher TA is that you have higher concentrations of acids, and in particular malic acid. Tartaric can be higher too, but malic has greater variation in the varieties we've looked at, and also higher mineral content, particularly potassium. So the combination of those two, more potassium, more malic acid. In other words, if you take a vinifera and somehow magically added potassium malate to it, you would make something that looks more like a typical hybrid. This is the root of many of the issues associated with producing wines from hybrids. You end up in a situation where you can't control the sourness, the TA, without getting the pH excessively high. So in the vineyard and the winery, this is a good, if you wish to correct pH and TA issues, first off, I strongly recommend, if you don't know what to do, talking to your local extension agent or attending a workshop. This is one of the most routine topics to find at a winemaking workshop or at an extension roundtable or something like that. When you have a situation like Vignoles or like many other interspecific hybrids where you have both very high, when you have both high pH and high TA, that's high mineral content and high titratable acidity, you are trying to address both problems, both the very high TA and the very high pH, by removing both acids and potassium, not just one or the other. For decreasing minerals, there's some approaches in the vineyard. One of the ways to have lower potassium is to have less water availability, if that's an option. I don't mention here, but not letting grapes get excessively ripe. One of the best predictors of high potassium is having very mature fruit from a sugar perspective. Another approach, what I call potassium fining, which is add tartaric acid early and then cold stabilize, that we'll discuss in the next part. And then you can also drive down potassium content even further by using cation exchange or electrodialysis. Decreasing acids, one of these is an approach you almost certainly are doing already, even if your goal is not to decrease titratable acidity, and that's to cold stabilize the wine so that you don't have potassium bitartrate precipitate in bottle. You can also decrease tartaric acid below equilibrium values through some other approaches such as electrodialysis. And then finally, for malic acid, in in the vineyard, it's a classic way of trying to decrease malic acid is to have well-exposed warmer clusters. And something else that I put in there is that you can also allow the grapes to mature longer. You can use yeast strains that will partially metabolize malic acid, malolactic fermentation, and in some of the textbooks, they suggest double salt. There's not a lot of great data to indicate that double salt's removing malic acid. It looks like this would be the addition of calcium carbonate in excess to a portion of the wine. It looks like most of what's coming out is tartaric acid when you do this, which still may be effective, but there's also other ways to remove tartaric acid as well. Okay, so that was a very brief summary of what could be a multi-hour workshop on managing pH and TA in a winery. So one of the things we're working on in VitisGen 2 is to understand the root cause at the grape level of what results in this both high malic acid in particular and higher potassium in non-Vitis species. And so what I'm showing here is a plot of the behavior of malic acid during the ripening of vinifera. And if you pick up a textbook on wines and grapes or on viticulture, you'll likely see a plot that looks something like this. Malic acid starts to accumulate in unripe berries. So when you start pea-sized berries here, they start to expand, grow, they're still hard and green, and malic acid is accumulating during this time. It's getting up to concentrations of 20 grams per liter, maybe 30 grams per liter. Right around or just before veraison, there is a shift where malic acid starts to be respired and possibly degraded in other ways as well. So malic acid will decrease for two reasons on a by weight basis. Number one is the berry is expanding further, so there's dilution. And the second is that malic acid is used as a metabolic substrate. It's respired, turned into energy and carbon dioxide and water. This is the textbook behavior for malic acid. This is for vinifera, because one of the things we've been working on more recently is what happens when we get outside of vinifera. And what we see when we look at wild vitis is, for example, here for Cinnaria and Riparia, which are two grapes of interest to grape breeders, is that on a per berry basis, malic acid does not decrease during ripening. In fact, in many cases, it looks like it continues to increase during ripening. And hybrids have an intermediate behavior between wild vitis, Riparia, Cinnaria, I don't show it here, but rupestris as well, and vinifera. And so, if anything, where we're seeing unique behavior, it's not the wild vitis, so the hybrids. The unique behavior is in the vinifera, or I should say specifically domesticated vinifera. We don't seem to find grapes in the wild that uniformly degrade malic acid in the same way that we don't find white grapes. In most places in the wild, we don't find grapes that have muscat aromas in the wild. These are things that humans have selected for. And it looks like one of the things that humans have selected for is a low acid phenotype for vinifera. So again, to summarize, these wild grape species, what we see is not a degradation of malic. We see, if anything, occasionally an accumulation or no change in malic acid per berry. The end result, why this matters to winemaker, is that these wild species and their hybrids will have less degradation during maturation, such that you can have a grape that is mature from a sugar perspective, 20% sugar, 20 bricks, and still has 20 grams per liter or more of titratable acidity at harvest. So one of the things that we're working on right now is to try to understand what genes are involved in the degradation or lack thereof of malic acid in these wild grapes as compared to domesticated vinifera. So we're working with breeders, especially one population in Geneva, that shows excellent segregation. That's differences within the population for malic acid behavior after veraison. And one of the other interesting things we're seeing is that this might be linked to uptake of minerals as well. And so where we hope to be in another few years is to have good markers for this behavior, as well as markers for malic acid accumulation that we can give to breeders so that they can start breeding more effectively for low malic acid phenotypes. Okay, so continuing on with challenges, low tannins in wine. So some quick terminology. Tannins are a biological polymer or a biopolymer. That is, they are big molecules that are produced by stitching together long chains of smaller molecule. And for example, in the case of cellulose, cellulose is made from a small molecule, glucose. And if you stitch together these glucose subunits into long chains, then you can make cellulose, and you can also make starch. If you change the types of sugars, you can make things like pectin. These are all examples of biological polymers. Condensed tannins are another type of biological polymer. They will not use glucose as their subunit. Instead, they use polyphenols as their subunit. So on the left, in the center, I should say here, this is a flavanol or flavanthriol. If you take this compound, which is a polyphenol, and make long chains of it, now you've made a condensed tannin, also called a proanthocyanidin. And these tannins can be, I say here, up to 80 units long. We've detected tannins with up to 80 units. Probably there's even longer chains present in grapes. They just become challenging to extract and characterize once you get to a certain point. So the common property of tannins, and there's multiple types of tannins that have been described, but the one thing they have in common is that they can bind to and precipitate protein. And so what all of these things have in common up here is that they contain protein, and in particular, all of these have proteins that will be important to a winemaker. If you have a wine that is excessively tannic, and so what astringency or tannic perception would be the perception of drying or the loss of lubrication in your mouth. So if you take a drink of something, like green tea or an astringent red wine, take a sip, you feel like your mouth is, you can't easily move your tongue across your lips or your teeth. Your mouth feels dried out. This is the perception of astringency. And just like hot pepper, like a lot of hot pepper isn't all that pleasant. A lot of astringency isn't that pleasant, but some of that is essential to what makes a full-bodied red wine feel full and in the mouth. And so winemakers will employ proteins from milk or proteins from eggs or from gelatin and other sources to do fining, that is to remove tannins from wines that are excessively astringent. And then in the case of saliva, you, the consumer, or the winemaker, if you're doing a tasting, you're using your salivary proteins to bind to and precipitate out those tannins. And that is what's leading to this perception of astringency. So these salivary proteins that combine tannin, these are the same salivary proteins that provide astringency, that provide lubrication in the mouth. So if you strip out all of your salivary proteins, say because you just consumed something very astringent, very strong tea, or dark chocolate, or a red wine, or a flight of red wines, you're going to have a perceived loss of lubrication in your mouth. You can also actually visibly see these interaction products. If you take a sip of an astringent red wine or green tea or something else like that and spit it out, over time you'll see some stuff floating in what you just spat out, and that's those precipitates formed by the reaction of proteins and tannins. Okay, so we want to get these tannins out of red wines, out of red grapes in many cases, and into the red wine. And one of the things that's well known anecdotally, but also from literature, is that vinifera wines, on the average, have higher concentrations of tannins than interspecific hybrid wines. So I'm showing up here first is tannin concentration in vinifera wines from New York State and from the West Coast and from Australia. On the average, warmer, drier climates accumulate more tannin in the fruit, and that tannin ends up being more extractable. So we see on the average more tannin in red wines from, say, California than the same grape. We've got Merlot compared to Merlot, Cab Sauvignon compared to Cab Sauvignon from New York. If we look at some of the classic interspecific hybrids and their resulting red wines, the only reason they're positioned here is I didn't want them actually off the computer scale because they should be at undetectable. This is Bac Noir, Liam Merlot, the Chanac, Maréchal Foch, classic interspecific hybrid red wines. These grapes produce red wines with undetectable concentrations of tannins, and the resulting wines are often described as thin or lacking astringency. So this is in part because hybrids on the average have less tannin, and in part because on the average that they tend to have less skin tannin in particular, but that's not the whole story. So we've done some work where we've looked at tannin in wines produced from fruit using exactly the same winemaking conditions, using grapes from New York State grown at nearby sites. On the average, hybrid grapes, shown here at the bottom, these are the classic French American interspecific hybrids, FA hybrid. They have, on the average, a little bit less tannin, but not that much less tannin. But when we look at the finished wine, we see much lower concentrations. In fact, in most cases, undetectable concentrations in these traditional French American hybrid grapes, and I should say in the wines produced from these traditional French American hybrid grapes. As a compliment to Bruce Reich, these are a couple of his newer releases, I believe, Noire and Cora Noir. We do see higher concentrations of tannins in some of those newer releases. So the problem with wild Vitis and with many of their hybrids is not just that they have lower tannin, it's that the tannin is less extractable as well. So what seems to be limiting tannin extractability is in large part the presence of tannin binding proteins. Instead of those tannin binding proteins being in our mouth, which is where we like those proteins to be interacting with tannins, instead they're in the grapes. And the highest concentrations we see of proteins in grapes is usually in wild Vitis or in their interspecific hybrid offspring. Put another way, you are doing protein finding of your wines when you ferment wild grapes, and often they're interspecific hybrids, except that those proteins are coming from the grape. So highest concentrations that we see are generally in Vitis riparia. Interestingly, Vitis cinerea, another wild grape, we don't see all that high concentrations of juice protein or wine protein. What we do see is very, very, very low tannin in Vitis cinerea. So in the case of riparia, it seems to have a little bit low tannin. The big problem that riparia and its offspring have is high concentrations of proteins as compared to many of the viniferas. Okay. So in the winery, what can you do? We don't have much in the way of viticultural work to provide suggestions. But in the winery, one of the things that we've worked on is, this is a protein. You have strategies that you can do in a winery for moving proteins. So one of the things we did was treating the free-run juice prior to fermentation. So you bring in grapes, you crush them, you free-run, rack that off, treat with bentonite, return. And you can get a little bit of improvement in the final tannin concentration. So control in the black versus the far right is bentonite treated juice. This is not completely effective because protein continues to be extracted from grape solids during fermentation. So this is only getting rid of the juice that's present in that initial free run. So if you're wondering, hey, can I just add bentonite to my fermentation? No, because the bentonite's still there, and bentonite bound to protein can still interact with tannin. I've talked to winemakers who do Sauniers and will take that free run and make a rosé from it and remove 30–50% of the initial juice and then ferment the rest on the skins. And the resulting wines, although we don't have any data yet, it seems like a reasonable approach to try to increase final tannin concentrations. You can add exogenous tannins. You can buy commercial tannins. If you do this, be prepared to add a lot more tannin that's recommended. most of the tannin you add is going to be bound up by those proteins. And you can also add tannin after fermentation. One of the problems with that is these tannins often have impurities in them that don't smell or look that great. They contribute a brown appearance. They have off aromas. So often by the time you've added enough of these products to create sensory difference in terms of astringency, you've created other undesirable changes too. And then you can also try to blend with a high tannin vinifera. It's one of the things, this is something I'm working on right now with a collaborator at Iowa State. This will work. It's another, same as exogenous tannin. You need to add a decent proportion of the vinifera to have an impact. And the biggest effects seem to be if you do the blending post-fermentation, presumably because you're getting rid of some of that insoluble or non-extracted protein when you do that. Okay, so in VitisGen 2, we again are taking the same approach as I described for malic. We're using mapping populations that show differences, that segregate for tannins and for tannin extractability. We have a collaborator who's an excellent population for this, using a vinifera, Cabernet Sauvignon, and a non-vinifera species. I mentioned this group before, our grape squishing team. We have a team of undergraduates and technicians that go through the dirty work, literally and figuratively, of transforming all of these individuals into samples that can be subsequently analyzed. And for tannins, we look both at how much tannin we have total. Right now we're looking at total tannin in the berry, although in the future we're going to be looking at that based on skin versus seed. And we're comparing that to the amount of tannin that actually gets out. And one of the better proxies for extractable tannin is to use something more wine-like 15 to 20% ethanol. And the difference between these, we think, is going to be a good proxy for the amount of protein that's in the grape. And again, what we like to do eventually is to provide markers to our breeder team so they can select for individuals that are going to eventually have higher tannin in the finished wines, both through having higher tannin in the original grapes and by having more extractable tannin in those grapes as well. One of the other interesting things that's come out of this project, and this was out of the first VitisGen, the predecessor to VitisGen 2, was you realize that there's some value to looking at these wild grapes because they make it easier to understand what's going on in vinifera. So I have a couple of other examples of extremes in phenotype where they've been helpful to researchers. For example, Thermus aquaticus, also called TAC, is the source of a key enzyme that's used in the polymerase chain reaction, or PCR, which is one of the standard approaches still for doing genotyping. And the reason it's of interest is it can tolerate extremely high temperatures. These will grow in deep sea vents underwater, and that property makes it useful for geneticists when they're trying to do DNA sequencing. Naked mole rats live underground, near blind. They, for reasons that are still under scrutiny, do not get most types of cancer. So these types of extremes, including Vitis riparia and other wild Vitis, are useful for understanding other situations where we don't have things that are quite as extreme. It's well established amongst growers of vinifera that grapes can differ considerably in their tannin extractability. For example, if you take Cabernet Sauvignon from multiple sites and vinify it in the exact same way, you cannot predict final tannin concentration from original tannin concentration in the grape very well. And what groups in Australia are now realizing is that what we learned on the protein side for Vitis riparia and for other wild Vitis, that seems to be occurring in Vitis vinifera as well, and can help explain some of this variation that occurs in vinifera too. So back to VitisGen 2, but part of the value of this project is not just helping grape breeders, it's also helping illuminate some of the hard to explain differences that we see within existing commercial vinifera varieties as well. Okay, lastly, the odor side and volatiles. So to characterize volatiles, we use a tool called a gas chromatograph, or GC. The idea behind GC is that it separates out volatile mixtures, in other words, a wine extract, which contains lots and lots of volatile compounds, some which contribute to aroma, into individual compounds. You can think of it as a distillation. only on a very small scale. We're separating out compounds mostly based on their boiling point. In other words, just like you would if you were doing a distillation. And so the data that comes off will look like a straight line over time with a series of peaks, and each of those peaks is a unique compound, and for GC specifically, it's a unique volatile. And then we can use other techniques to identify what those volatiles are, for example, a mass spectrometer. One of the challenges with doing aroma analysis, and this isn't just for grapes and wine, but for a wide range of foods and beverages, is that just because something is volatile does not mean it has an aroma. Or just because it has an aroma doesn't mean it has an aroma at the concentrations where it's present within grapes or within wine. So for example, we have this big peak here corresponding to phenylethanol in wine. And its concentration is roughly proportional to its peak size, which is around 10 milligrams per liter. And then we have this other, I can't call it a peak because you can't see it. You actually have to use a different technique with more pre-concentration and separation to see it. But trust me, if you squint hard enough, maybe you can see a little peak there for a compound called IBMP, or isobutyl-methoxypyrazine, which has the smell of bell pepper. Its concentration is 1,000,000-fold lower than phenylethanol. So based on concentration alone, it looks like phenylethanol should be the more important compound. But just because something has a high concentration doesn't mean it has high potency. It doesn't mean it has a sensory effect. And if we compare these concentrations to their sensory thresholds, one of the things that we learn is it's often low concentration compounds, for example, IBMP, or someone 4-MMP, which has the smell of cat pee or boxwood, that are the key contributors, in this case, to Sauvignon Blanc, while other things that are present at high concentrations are nearly irrelevant, like phenylethanol, even though it's present at orders of magnitude higher than IBMP or 4-MMP. So one of the things that we've done in our group is to use GC mass spectrometry, which can help us identify compounds and quantify them in combination with GC-Olfactometry, where a detector at the end of the GC is a human being sniffing what comes out of the gas chromatograph. They are only going to be sensitive to things that have a smell, and a smell of the concentrations that are going to be where they're present in the original extract. So using these tools, our group and other groups have been able to identify some of the compounds that contribute to wild vitis that are present at much lower concentrations in vinifera. In the case of Labrusca, there's a methylenthranolate, which has been known for some time to contribute to that Jolly Rancher grape Concord-like type aroma of wild Labrusca type grapes, and O-aminoacetophenone, which has a more like a Niagara grape-like characteristic, and not shown here, perennial, which smells like cotton candy. And in the case of Riparia and Rupestris and Cinerea, we've identified several compounds, including methoxypyrazines, which I mentioned before, have a green pepper vegetal aroma, and other compounds that have vegetal and herbaceous aromas. So if you want to limit foxiness, or that Concord grape, Niagara grape type characteristic in a winery, one of the strategies is, and this is something that's widely used already, is harvest early, real early. For example, I know that people who are using Niagara and Cayuga White and Delaware who want to try to avoid or limit foxy characteristics in the resulting wines might harvest, I've heard of Cayuga White being harvested at 11, 12 bricks, so as opposed to the more typical 16 to 20 that you might see for most wine grapes around where we are. That's one way to try to limit foxiness. Another approach that we're working on in my lab right now, pardon me, you probably don't care too much about the news update, do you? Although you might, if you're in front of the computer, feel free to go Google New York Times or some other source if you're curious. Other possibility we're working on right now is, can we selectively remove some of these foxy odorant compounds before fermentation? Most wine odorants aren't present in the grapes. They're formed during fermentation. Foxiness is an exception. So can we use juice treatments like nanofiltration or vacuum distillation to selectively remove off aromas from the grapes or the wine, and then ferment, and then we end up with something that smells more like a neutral red or a neutral white wine. We've got some initial results indicating that this is a possibility. Okay, another approach you can mask what's there. And this is not uncommon for people who are producing wines from interspecific hybrids with small berried vitis in their background. So some of the classic French-American hybrids are often treated this way, like Maréchal Foch, which is that you use techniques that will give you very rapid color extraction, and then you press, and then you ferment like a white, rather than skin fermenting to completion. When you do this to red grapes, when you press early and following some sort of aggressive extraction to get color out, and you ferment without skins and without oxygen, this stimulates production of acetate esters. These are some of the key aroma compounds found in Beaujolais Nouveau and several other fruity red wines. And there's a lot of ways to achieve this. Carbonic maceration, Beaujolais Nouveau will do this, thermovinification, flash détente, which is a variation of thermovinification. Rotofermenters, to some extent, can achieve this. You're still going to have some of the off-aroma compounds from those red grapes, but they'll be masked, and if it's designed to be a fruity, early released red wine, this might work effectively. So again, on the VitisGen 2 side, we're taking a similar approach to trying to characterize off-odorants. We've got multiple sources of vitis, interspecific hybrid, of wild vitis-based mapping populations. We are characterizing a panel of around a dozen or so off-odorants, depending on the population. And then we eventually hope to work with breeders to provide targets for them that they can use to to screen out undesirable characteristics early. But one last point I want to make is, are we making a mistake to think that these always need to be off-aromas? I don't think there's necessarily a lot of value to having grapes that routinely have 5% malic acid by weight. Maybe there's a niche application. But one of the things that makes wine is its diversity. The fact that we go to the wine store expecting that all those different bottles are going to deliver different sensory features. And this has been the reason that we have selected for, over centuries, varieties that provide unique sensory and particularly unique odor characteristics. For example, Gewürztraminer grapes are characterized by having high concentrations of cicerose oxide. Muscat type grapes, so Muscat of Alexandria or Muscat Valvin are characterized by having high concentrations of linalool and geraniol, which have floral type aromas. In the case of wild vitis, especially the large berried wild vitis, we see high concentrations of foraniol. This may be considered a fault in a lot of wines, but in the case of table grapes, such as the new cotton candy varieties, this is a case where the diversity was celebrated, and it made for a new table grape variety that you can find in supermarkets. If you look at the volatile profile of some of these small and large berried vitis, you can look at them from two ways. One is that anything that doesn't look like typical landscape of vinifera is a problem. The other is to look at it as an opportunity. For example, there are small and large berried wild vitis that have high concentrations of eucalyptol, minty aromas. And this has been, you can find these actually in Himrod grapes, which is one of the classic Geneva table grape varieties. You can find many wild species that have high levels of eugenol, although it's often masked by some other terrible smelling compounds. And there's dozens of other interesting compounds that should be able to be retained during fermentation, be stable in wine, and potentially deliver unique sensory characteristics. And maybe this becomes part of future cultivars, not just getting rid of things we don't want. Okay, so to summarize, since we're almost out of time, wild vitis and some of their interspecific hybrids can present flavor challenges. The advantages are cold hardness, disease resistance, and other good growing properties. The challenges are excessive sourness, low tannins, low tannin extractability, and off aromas. So those are the three areas that I highlighted today. There's some existing viticulture and winemaking practices that can mitigate these issues. Many of the newer hybrids that have been released either don't have these problems or have less of these problems, but there's still interest in trying to become more efficient during the grape breeding process, that we can, and the grape breeders can bring online new varieties that deliver desirable flavor, but also retain the properties of wild vitis that we want. And finally, wild vitis might not just be an issue of trying to get rid of all of the flavor issues. It may present an opportunity for new types of flavors in new cultivars as well. And I think this is a good time for me, first off, to acknowledge lab members who've contributed some of the data shared today. On the left, Noam Resheff and Beth Brzezinski have done much of the work on the malic acid side. Heather Scott has overseen a lot of the general chemotyping, especially oversight of what I refer to as our undergrad grape smushers, smushing team, the people who are doing the characterization of all of these mapping populations. Our funding source is the USDA through Specialty Crops Research Initiative. And finally, I'd like to thank the larger, broader VitisGen 2 team, particularly Tim Martinson and Janet, Janet's name is not up here. I'm so sorry. Van Zoren, who's not up here, unfortunately. Thank you for inviting me to participate today. And if there's time for some chat-based questions, I think I can try to answer them.
Great, thank you, Gavin. That was really well organized and very comprehensive. So we do have time for a few questions. So if anybody wants to type them in, just put them in that Q&A.
Sure. And should I go to the chat myself? Or are you going to?
I'll just ask them to
okay.
you. So far there aren't any in there, but I'm guessing some people are typing in. But in the meantime, while we're waiting for other questions, I'm not really sure this is a question per se, but I was sort of, as you were talking, thinking about these different concerns that you're talking about in terms of TA and tannin concentrations and off aromas, and just sort of how the breeding and sort of vineyard management and how they're treated in the winery as well as marketing, how those sort of all play into these concerns, and wondered if you might talk briefly about sort of like how you see those playing together, or if any of them you think should take a stronger role.
Oh boy. Okay. So
that's pretty broad.
So the
Sorry.
things that I have to address are breeding and marketing, and
and then sort of like vineyard practices and winery. Like
yeah. So the vineyard and winemaking practices. That's so there's the short term and then the long term issues. The short term issues are if you're trying to breed for new varieties, that's a long-term process. It can take years, if not over decades, to go from making a cross, evaluating with grower cooperators, doing sensory trials. We hope to expedite that process, but if you want to know what do I do right now while waiting for that, you're going to be working with existing cultivars. And planting something that makes sense for where you are and where you're growing things. And that's where viticultural decisions and winemaking decisions make a lot of sense. And it's good to work backwards from what can you achieve, what do you want to make, and then to think through whether or not you're actually going to have the grapes available from where you are to achieve that kind of product. You can also go the other way of what grows best in terms of like from a viticultural side, and then say, what can we make from this? So on the marketing side, one of the things that's nice to see in the market, and one of the things that still has been a handicap, is that consumers in the United States and in a lot of other New World regions were varietal focused. That's how they selected wines. I want a Merlot, I want a Cabernet Sauvignon. That still happens to some extent, but the fastest growing sector of the red wine market, Cabernet Sauvignon is still doing pretty well, but if you look at the data close to it, and sometimes besting it, is no particular variety. It's red blends. That is the fastest growing part of the wine market. White blends are there as well. At the national level, there's a number of popularly priced red blends that are doing very well. For example, Gallo is a product called Apothic Red. What grapes are in it? Well, you can look and you can find out, but that's not what's selling the product. And here on the East Coast, the Finger Lakes, I know we've got a number of winemakers that are doing some very successful blends. And it's nice to see consumers becoming more interested in those types of wines, non-varietally labeled wines, and that's one of the opportunities for these new cultivars.
Great, thanks.
Can I jump in with one here real quick?
Yep.
Gavin, if your goal is to, as a breeder, is to emulate something that would taste like Cabernet Sauvignon or Pinot Noir or Riesling, are markers going to get us there? Are we going to be able to identify markers that will sort of get us there or closer?
So it's easier to talk about, very generally in flavor chemistry, it's always easier to talk about faults or problems than it is to talk about what makes something it gives something its underlying character. So as an example, if you were to give me a Cabernet Sauvignon, another Cabernet Sauvignon, and say, hey, one of these smells like moldy basement, the other one doesn't, but I'm pretty sure they've got everything else the same in them, it's pretty easy in most cases to identify what the cause is. Probably you've got some of the compounds associated with cork taint, because you've got, in that case, you've got one or two compounds that are different and that are creating everything about what makes those wines different. If you're talking more generally about, I want to make something that's Cabernet Sauvignon-like, then it's not just a matter of getting rid of all the things that make that wild Vitis clearly different. You actually need to try to match a broad range of chemicals to Cabernet. So where I think you can get to is, say, making something that people, if you have the broader category of is it vinifera-like? Can people tell that this wine produced from this interspecific hybrid is an interspecific hybrid or a vinifera? And you have them do an evaluation and like have them like guess or evaluate a panel of wines. I think these are vinifera, these are interspecific hybrid. You can most certainly, I think, get to that point where you have those cultivars. It's going to be far harder in most cases to try to duplicate some of these other varieties exactly because you're trying to match a lot of things. The exception might be where you've got cultivars where you've got one or two things that are in gross excess, and that creates the character of the wine. Muscat type grapes kind of go into that category. And Muscat Balvin, when it's well made, that's an interspecific hybrid, it becomes hard to tell that apart from Muscat of Alexandria.
Thanks, Kevin.
But once you get to Pinot, yeah, it's gonna be a little more challenging for some of those other varietal wines that you just described. But I'm not sure if it's necessary. I think that you're more looking to try to get things into the sphere of vinifera-like. You're not necessarily trying to make it just like Cabernet.
Great. We have some good questions coming in. So one, a couple questions about tannins. So Bruce was wondering, should breeders be paying attention to lowering protein or increasing tannin levels? And also on top of that, are we likely to find a single gene controlling these traits or multiple genes?
Right. So the protein part, let me go for both of those. We have seen considerable environmental variation in protein with some of the mapping populations we've looked at, more so than tannin. So I suspect, just from a breeding perspective, it's going to be easier to identify stable markers for tannins. That doesn't mean the protein part's not. It doesn't mean it's unimportant. Just it's going to be easier to get to the tannin side. So the general question of which is more important. My suspicion is that if you have varieties where you have enough tannin, you can probably outweigh the effects of low extractability due to high protein. And we know that already from spiking experiments. You put enough tannin into a wine during fermentation, yeah, you can end up with higher final tannin. So that's my guess is on the long term, while both might be important, if I only had to pick one or the other, I would say let's try to breed for tannin. I suspect it's going to be easier to find markers for. With that caveat, we can do an assay to try to characterize both tannin and tannin extractability. We're going to do that and see where it leads to, and try to validate with some small-scale winemaking trials as well.
Great. We have a few more tannin questions while we're on the subject. So Lou at Minnesota is wondering about the inheritance of tannins and whether hybrid progeny of riparia with vinifera would have improved tannin concentration.
Yeah, so from the little bit that we've done, because there's not a whole lot of populations that are based off of say only a single vinifera by a lower tannin, a wild vitis, or where the parents were intentionally selected to have high tannin, low tannin. As an example, there's a population in California that Andy Walker set up that's Riesling by Cabernet. Riesling's got plenty of tannin. It's just that we don't usually know that because we don't skin ferment Riesling. That's not a great population for looking at high tannin versus low tannin. We want something where we have big variation. From the one population that we do have that we've looked at, we do see considerable segregation in the trait. So my guess is that there is inheritability, but it's still premature. I understand when people have used, in a little bit, I've talked about people who've used other high tannin grapes in breeding populations. They do see some segregation in the offspring, but I don't yet have anything quantitative to share to back up that statement.
Okay, and then another question, whether you know anything or whether there's any research about using either stems or seeds and the tannin concentration in those, whether that could be used to increase tannin content.
Yeah, the challenge with stems and seeds is if you're putting those into a fermentation, yes, they have tannins. That is not a selective winemaking treatment, by which I mean you're going to have other changes. So sure, that's a source of tannin. And it's also a source of, among other things, green aromas too. And it can also potentially be a source of protein. When people add adding seeds or other material other than grape to their ferments, yeah, you can see a small bump of tannin, maybe even see a large bump of tannin. I'm basing this off of vinifera studies. You'll also see other sensory effects. That's not always desirable. So sure, that's a strategy in addition to using, say, a higher tannin grape or for blending or using exogenous tannin. But you should evaluate whether or not you can achieve a good impact on the mouthfeel while avoiding undesirable effects on other characteristics.
Great. And then one final tannin-related question from John asks, it has been proposed that many of the tannin-binding proteins in hybrids may be proteins that are important for disease resistance. Is that just speculation, or is there data to support that?
Yeah. So one of the problems, these are called pathogenesis-related proteins, and these are the same proteins aside as the ones that people who are producing white wine would remove by bentonite fining to prevent protein haze. So these are not unique to reds or unique to interspecific hybrids or wild vitis. The difference is that they're at higher concentrations on the average in wild vitis. So that terminology, pathogenesis-related proteins, and some of the names of these proteins, chitinase, beta-glucanase, they represent a time from when they were discovered, not in grapes, but elsewhere, when it was thought that they were, that their only role was in disease resistance. It looks like they do have some sort of role in signaling pathways associated with the incidence of disease. I believe Bruce Reich has done some work where they tried to upregulate production of these pathogens-related proteins, and they saw no difference in disease resistance. I don't know if Bruce is on the line, if you can comment. My understanding is that it's thought these are involved in signaling, but just like a fire alarm doesn't help you all that much just by, once that's a certain volume, increasing the volume of the 10 decibels doesn't make the evacuation process go any better. Once you have a critical number of these proteins, it seems like the signaling pathway is there, but they also have other roles that are identified as well. They seem to generally accumulate during grape ripening. They accumulate more when you've got abiotic stresses like high temperatures, like low temperatures and cold events. So just crediting higher concentrations to disease pressure alone doesn't seem to be accurate. There seems to be a number of other abiotic and biotic stresses that can result in higher concentrations. It also then looks like the upside is you can have lower concentrations and very likely not affect the disease-resistant properties of the grapes once they're bred.
So Bruce is on the line, and he just sent me a message saying that he saw some increased resistance to botrytis under certain scenarios, but little effect on other diseases.
Okay, thanks,
so
Bruce.
There's the answer. And then kind of moving back to what you can do immediately in a winery, Anna was wondering whether you could just briefly describe some of the best immediate strategies for winemakers in states where there's a lot of these interspecific red hybrids, such as the Midwest, Minnesota, Wisconsin, Michigan.
Wow, this sounds like a fantastic idea for another workshop. So, but you said briefly, right? And so the question is, how can you make great wine from these hybrids that you have right now? Was that the question?
Yep, exactly.
Or so to start off at the very high up level, what are you trying to make, and is it realistic? If you survey the landscape of other people who are producing things in the areas around you, and they are not able to achieve whatever kind of wine it is that you're able that you want to make, chances are this is going to be hard. So if you want to make a very big, full-bodied red wine, highly extracted, that is hard to make right now with not just with red hybrids. It's hard to make that with vinifera. In our region, due to other climatic issues, for example, lots of rainfall. So I think one of the starting points is, what are your expectations? What are you trying to make? And is it appropriate and realistic? The other big things that I would be concerned about are a lot of the standard viticultural practices that are recommended for red vinifera seem to have some similar effects, even if we're not always sure why, on interspecific hybrids. For example, here's one where we know why. If you have well-exposed grapes, you'll have less disease incidence. That's good. Even with interspecific hybrids, you can still have disease pressure. You'll have warmer grapes that will result in usually better color accumulation, but also less malic acid. For reasons that we're not entirely certain of, you end up with less herbaceous aromas in the resulting wines. This we don't think is methoxypyrazine related, because we see this even with interspecific hybrids that don't have high methoxypyrazines. But I've done some work with Justine Van Hoovel showing that using cluster thinning and using leaf removal and shoot thinning and other practices to open up the canopy, you can improve the resulting qualities of the wines if what you're after is less green aromas, lower sourness. In the winery, it sure is nice to know where you're starting from chemically. So there are benchmarks already for vinifer grapes, for acids, and for minerals. There's that information available for some interspecific hybrids as well. Where are you? If you're having problems with pH and TA, figure out where you're starting from, figure out what acids are contributing to that, and then you can use that to try to come up with a logical plan for getting your pH and TA in balance. Excessive sourness, it may not be the most exciting thing to talk about, but excessively sour red wines and white table wines are not pleasant. And trying to do all sorts of other things to the wine, if the sourness is out of balance, then the wine is going to suffer. So those are some of the basics. Is where are you chemically? What problem are you trying to solve? Too much malic, most likely. Very high minerals, very high tartaric. That's a little bit less common. Figure out what your struggle is, and then you can use that to define an appropriate way to move forward for the big basic thing, which is pH-TA balance. I think we could keep going from here, but I'm concerned that this could take a lot of time. But I think those are the big obvious things that I would focus on. What are you trying to make? Is it something which you can realistically achieve? Are you using good standard viticultural practices that have been recommended for producing high quality wines? And are you managing your pH and TA balance?
Great, thanks. Yeah, that's a pretty broad question and a lot more to explore there. We have a couple more questions already typed in. Do you have time to answer?
I have time right now. Yep.
Okay, perfect. And also just a quick note, if you have your hand up, if you're listening in and you have your hand up, I can't call on you. So if you have a question, just make sure to type it in to that Q&A bar. Thanks. So another question or kind of a comment is one of the most common characteristics of Frontenac red wines is a strong black cherry aroma, which is actually considered pretty attractive in most cases. And so the question is whether that's characteristic specifically to its riparia parentage and whether you can use that to kind of blend in that desirable characteristic to other wines.
Yeah, I do not, am I allowed just to say I don't know? Understand as a broader point, Pinot Noir is associated with cherry type aromas, often red cherry, occasionally black cherry. There has been dozens of studies to try to understand what leads to cherry type aromas in Pinot. None have come up with anything conclusive. There's some list of compounds that correlate, and for the most part, things don't make any sense. So if that's the same case with Frontenac, that doesn't mean that there's not a characteristic aroma for Frontenac, but it does mean trying to find a good chemical target for that characteristic, which might be an emergent property of a number of odorants, could be really hard. So yeah, it could be something which could be selected for, but it's something where the selection process might have to happen at the sensory side, which is a longer, slower process than at the chemistry side if we don't have good, robust markers. I know Anna Caston's done a little bit of work, and I've seen some other reports. When I look at what I've seen from the data is nothing that suggests something that would be unique and distinctive about why Frontenac would have black cherry and other grapes or other varietal wines would not. So yeah, it could be used, but the hard thing is finding a robust chemical marker for the trait that you care about. Not just, as an example, finding, I'm not sure, isoamyl acetate or an ethyl ester that sure has a fruity smell, but we find that in lots of wines at really high concentrations. What's unique about Frontenac? So this doesn't mean it doesn't exist. It doesn't mean it couldn't be found. We just don't have it.
Interesting, thanks. And then one final question, will enzymes as an enological product be able to assist in the extractability of tannins, aroma, color, etc., specifically for those hybrid varieties?
Yeah, so let's talk about, because the tannin one is where the main extractability issues come up. Anthocyanins, to the best of my knowledge, we haven't seen major issues with anthocyanin extractability. There can be issues with long-term stability of anthocyanins if you don't extract tannins as well. But let me focus on the tannin side, because that's where the extractability question definitely comes up. So we've looked at some enzymatic treatments, as has Anna Catherine's lab, I think some other groups too. One of the things I stress to my students, if any of you are listening right now, this will sound familiar. Whenever we talk about treatments during red wine maceration, there's two things you're thinking about. The kinetic effect, that is how quickly does the extraction have, and the equilibrium effect. Where do you end up given a long enough time? Enzymes routinely affect the kinetics, how fast you extract things. They tend to have less effect on final concentrations and equilibrium. So where these enzymes can make some sense is if you're planning on pressing off quickly. They will have a small effect on final concentrations of tannins, but a pretty small effect. And so why would this be? Well, if you use a pectinase, and you're trying to, if we see like the grape cell as a balloon, that pectinase is a pin. It's popping the balloon in a few places and allowing things to come out. It is not popping every single bond between a rubber molecule and another rubber molecule that's present in that balloon. It's not turning it all into monomers, which means that if you're getting, if your cell wall material is binding your tannin, that can still bind up. The other thing is, the tannins are not the only thing that's present in the grape cells. Proteins are there too. So if you use a pectinase, not only are you making tannin extraction more rapid, you're also making protein extraction more rapid. And this was not work that we did. It was work by that Australian group that had looked at proteins in Shiraz, and they showed that with pectinases, it just got them to the same endpoint faster in terms of final tannin concentrations. So sure, you're extracting tannins, but you're also extracting proteins at the same time. And one of the interesting questions is whether or not we can put to use something like proteases or some other more selective approach during fermentation to try to get rid of those proteins, but not touch the tannins. That's an open question. getting enzymes to work during fermentation is not easy. And that's something that's it's most certainly worth a look on the research side.
Okay, great. Thank you, Gavin.